Imagine for a second being able to see directly inside a whole organ, like your brain, maybe your heart, but without actually, cutting it open at all. Picture all those tiny, complex structures just becoming clear, almost like they're made of glass. Sounds, well, impossible, right?
It really does, like
Yeah.
something out of a sci-fi movie. But that's what's so incredible here. This isn't some far-off idea anymore. It's actually happening now, thanks to this really groundbreaking technique. It honestly looks set to completely change how we study biology.
Okay, let's get into this. We're doing a deep dive today on a pretty stunning breakthrough. It's from researchers in China. It's published in Cell, which is, a top journal. They've figured out a way to make biological tissues, even whole organs, totally transparent. So our mission here is to unpack how they're pulling this off, what makes it work, and, well, what it really means for understanding these incredibly complex systems inside us.
Exactly. The core idea, the real benefit, is letting light penetrate deep inside, exposing all the inner workings with amazing resolution.
So you can see tiny interactions without slicing things up, preserving everything as it is.
Precisely. No dissection needed. You get the whole picture intact. It's like finally seeing the entire building's wiring diagram instead of just looking at one floor or one room even. You see the system.
Right. It feels like magic. But how are they actually doing it? What's the trick to making, say, a whole brain see-through?
Yeah, that's the core of it. The key is this new method they call VIVIT. Now, the full name is a bit of a mouthful, vitreous, ionic, liquid solvent-based, volumetric inspection of trans-scale biostructure.
Whoa, okay. Let's stick with VINT.
Definitely. What it does is use these special compounds called ionic liquids, ILs for short. These aren't your everyday solvents. Think of them like salts that are actually liquid at pretty low temperatures. And they are absolutely central to this whole thing.
Ionic liquids. What makes them so special? I mean, people have been trying to make tissues transparent for a while, haven't they? What were the old problems?
Oh, absolutely. It's been a challenge. Older methods often used organic solvents or water-based solutions. And while they could clear tissue, they often caused problems, like shrinking or sometimes swelling.
So they distorted the very thing you wanted to study accurately, like
Yep.
trying to map a city with rubbery streets.
Exactly. That distortion was a major hurdle. But this is where the ionic liquids in VIVIT are, well, revolutionary. The researchers found that when you treat the samples with these specific ILs, something called vitrification happens.
Vitrification.
Yeah, it essentially turns the tissue into this stable, glass-like state. And crucially, it does this without forming damaging ice crystals when it's cooled.
So it's like freezing it perfectly solid, but in a way that doesn't wreck the cells. Protecting the structure allows light through. That non-destructive part sounds key.
That's precisely it. VIVIT preserves these incredibly delicate samples perfectly, so scientists can now look at structures across huge scales, from the whole organ down to, the finest nerve connections, without damage. This totally changes things for high-res microscopy. Usually, you need thin slices, often damaged now. You can potentially image the whole intact organ.
Okay, that's transparency sorted. But you mentioned this was surprising. Did they find anything else? Any other, unexpected bonuses from using VIVIT?
They did. Two really cool things, actually. First, totally unexpected, VIVIT massively boosts the signal from fluorescent dyes.
Boosts it. How much?
By up to 38-fold for certain dyes. Imagine trying to see a tiny light bulb and suddenly someone turns on floodlights behind it. That's kind of the effect.
Right.
Much clearer, deeper imaging.
Wow, okay. 38-fold is huge. And the second thing?
The second was a clever software they had developed called TARRS, a Trans-Scale Acquisition, Reconnection, and Reconstruction System. So even if you do need to slice up a sample for some reason, TARRS can take those images and digitally stitch them back together into these really precise, high-fidelity 3D models. It complements the whole organ view.
That's amazing. Stitching it back together virtually. Okay, let's connect the dots. What does all this VIVIT, the transparency, the signal boost, TARRS, what does it mean for understanding how our bodies actually work? What new doors does it open?
Well, that's the exciting part. It's not just making current research easier, it's enabling entirely new questions. Think about the brain. Now you can do precise 3D mapping of how neurons connect across large areas, like how multisensory neurons in the thalamus, the brain's big relay station, send their signals out across the entire brain, that kind of map, essential for understanding complex functions, or even dysfunctions.
So getting a real system-level view.
Exactly. And they're also gaining new insights into something called inhibitory control in the human cortex. Basically, how our brain puts the brakes on activity, which is fundamental for, well, everything. processing information, focusing attention.
Okay, so wrapping this up, VIVIT lets us make whole organs transparent, preserves their delicate structure, massively boosts fluorescent signals for better visibility, and helps create precise 3D maps. It really seems to overcome those old limitations of distortion and destructive slicing.
Absolutely. It's giving us this unprecedented window into the body's hidden machinery, helping us untangle those complex 3D networks, especially in the brain. It's about understanding the blueprint of life in a way we just couldn't before, getting us closer to maybe solving some really big biological puzzles.
And here's something to think about as we finish. While this VIVIT tech is clearly revolutionary, the researchers themselves note, they still need to figure out exactly how these ionic liquids work their magic. How do they interact with the tissue at a molecular level? How do they actually amplify those fluorescent signals so much? So the question is, what new discoveries, what even more powerful techniques might emerge once we fully understand the why behind this amazing transparency? We've got the window now, but figuring out how it's built. That deep dive is really just starting.